low-density lipoprotein receptor can be found in liver cells where it can perform its function efficiently. The LDLR starts with the LDLR gene in chromosome 19. This gene provides the instructions for making a low-density lipoprotein receptor. From chromosome number 19 in the nucleus, the gene is transcribed into an mRNA. Then the mRNA travels out of the nucleus and into the rough endoplasmic reticulum where the protein is synthesized by ribosomes. The key thing to remember about this receptor is that it is a lipoprotein, which is a protein with a component of fat. So after the protein is synthesized in the rough endoplasmic reticulum, it heads through the smooth endoplasmic reticulum where the fat component is synthesized. Then a transport vesicle takes the lipoprotein to the Golgi body where it is packaged in its respective shape of 2 alpha helixes and the rest of its structure made of beta sheets. For the sake of this model, we will be using a paperclip to represent the receptor. The receptor goes into another transport vesicle where it is taken to its final destination, the cellular membrane. Once it is embedded in the membrane, it serves to regulate the amount of LDL cholesterol, a low-density lipoprotein. It recognizes the apoprotein B100 that is located in the outer phospholipid layer of the LDL particle and binds to it. The LDL receptor particle complex, which is the name of the structure when the receptor and its particle are bound together, then goes through endocytosis, a cellular process that involves a part of the cell membrane to butt off inside to bring whatever that is outside of the cell inside. Once the LDL particle is inside the cell, a lysosome engulfs it and then degrades it. Once it is broken down, it releases cholesterol, which is used by the cell, stored, or even removed from the body. When the LDL is dropped off, the LDLR is recycled back to the membrane to bring more LDL. The reason why LDL receptors can most commonly be found in the liver is because the liver's function is to filter blood, and the LDLR can help by taking in LDL cholesterol. Because the LDL receptor plays a significant role in the liver, it's important to know how quickly cholesterol can be removed from the bloodstream. This factor is a way to identify the many mutations that can occur. There are 2,000 mutations that have been identified in the LDLR gene that can cause a form of familial hypercholesterolemia. These mutations can reduce the number of LDLR produced or disrupt the LDLR's ability to remove LDL. As said before, the normal structure of LDL receptor includes 2 alpha helixes and the rest are beta sheets. In one type of mutation, the receptor loses a link between one of the 2 alpha helixes and a beta sheet, which damages the ability of the protein to function properly. As a result, there are high levels of cholesterol in the blood. The excess cholesterol is deposited in places where it shouldn't be, such as tendons, skins, and arteries. A buildup of cholesterol in the arterial wall can cause a disease called atherosclerosis, which causes the blood flow to be inhibited. In a healthy artery, there are no blood clots, so a blood cell can easily pass through. But in one that has atherosclerosis, there is a blood clot formed, and that makes it harder for the blood cell to pass through easily.